This is lab 11. Echinoderms, hemichordates, urochordates, cephalochordates. I have a PowerPoint. And we will stay in this application. Okay, so here we are, echinoderms, urochordates, hemichordates, cephalochordates. And this is where we are, what we are looking at today on the tree of life. So this is not a monophyletic group that we are looking at. We're checking out these animals, of which there are very few. They are not vertebrates. We are vertebrates. They are chordates. We are chordates. So this is one of those creatures that if you you could flip this whole, I'll show you, you could flip this node on the tree around, and then these guys, the urochordates, would be way out here, and that could potentially make a little more sense to some people. Remember, these nodes can be flipped. It does not change the relationships between the animals, only the way it's visualized. And that is important to understand, because if you don't understand that can be flipped, then that means you don't understand what a phylogeny is. So important to know. So this is a little bit of recap. Up until right this moment, we have not entered the realm of the deuterostome. We've talked about some protostomes, and we've talked about animals that are neither protostome or deuterostome. So this is complicated. These are loci on the genome that we've done some experimentation on, that has allowed us to identify where some of the split occurs, like where the differentiation is. We can see it in the sequence, and some of it has to do with Hox genes, and Hox genes shape the directionality of cell movement and cell differentiation during development. So that's a little bit of background, but here we are, stepping away from this for a moment and entering the realm of the deuterostome. And part of doing that is recognizing what we're leaving behind, what the differences are. So this slide, I feel like I keep showing you versions of this image over and over and over again. But what I haven't talked to you about yet, I barely touched upon it, and I remember when I did. This difference here, so we've got 3 triploblastic layers for protostomes, which include everything from last week, and deuterostomes. They all have ectoderm, mesoderm, and endoderm. Ectoderm, mesoderm, endoderm. But look how the mesoderm forms here. The things that have a gut, and where the muscles are at. The muscles, skeleton, most connective tissues, that's mesoderm derived. Here, and that forms from it either forms from a pocket, or it's an out pocket. So it's a pocket, self-generating pocket, or it's an out pocket. And the out pocket is a pinching type of generation. So that's what happens here with the deuterostomes. You have radial cleavage, which is the most basic kind, the easiest to visualize, and then you have a blastula. Then that thing inverts. It basically swallows. One instructor phrased it to me as it's like your upper lip going over your lower lip and then swallowing your whole body. That's sort of what happens during gastrulation. And post gastrulation, there's some dimensionality. And in the deuterostomes, that little hole where the swallowing began becomes the anus. So the anus is first. This is the blastopore. It's the anus. Told you that a lot. Deuterostomes are anus first. Protostomes, pro first. Hello, stone. No. Deuterostomes different. Second mouth. Second opening is the mouth. Anyhow, so these orange cells are ectodermal, and here the yellow cells are endodermal. Pretty intuitive. It's very much like a C, and the inner lining of the C pockets off to form the mesoderm, rather than cells around the ectoderm, like where becoming mesoderm cells. So cell fate of these three basic layers from which everything you wind up with is derived, and this is called enterocoely. Let's see if I can. I don't have my special little writing glove today, but you know what? Actually, it shows up in the next slide, or I've got a slide coming up that spells out the word for you. So enterocoely is how the mesoderm forms with deuterostomes. Okay, so going back to that tree, the cephalocordate part of the tree that we're looking at today. Here's an example of it. So this is called Hycoella. So it's a chordate, and it's a fossil. I should mention that this is I think it's Precambrian. No, it's either Cambrian or it's probably Cambrian. I'm not sure. It's old, like more than 500 million years. So it had a filter feeder situation, something called an endostyle, which secreted enzymes and mucus, and it's a precursor for what evolved into the thyroid gland in higher chordates. So early on in our development, we have something that resembles this thing called an endostyle, and it becomes more complex, differentiates, produces a lot of hormones, and also powerful enzymes, which it does here. So the endostyle probably had some maybe some immune function. Usually where enzymes are secreted, foreign molecules are taken apart. So this could have protected the animal a little bit and also digested food as it was going in. So they had a nerve cord, a dorsal hollow nerve cord, and it had a notochord. So guys, at some point, probably on the lab practical, definitely in a quiz, definitely in lecture, there's no escaping you having to know that all chordates have a notochord. You need to know that the notochord is not the same thing as a spinal cord or the vertebral column. And you also need to know that where it is in respect to the dorsal hollow nerve cord. So I'll show, as this PowerPoint progresses, I'll show you some ways to recognize that. But notochord and dorsal hollow nerve cord. Also, now we have myometers. So earlier, I've shown you longitudinal muscles and circular muscles that are nearly always present in the same animal, like a worm. The earthworm, Lumbricus terrestris, is actually a very muscular creature, and so is Chimbaris, the crayfish. But now we have something, and there even spiders have quite a few muscles, but these are big, long, thick slabs, and when they contract in tandem, they can have a side-to-side swimming motion. So this is how the first early chordate swimmers, cephalochordate swimmers. We know that as best we can know something that we can't see without a time machine. That the first of those swimmers were not undulating like a wave, but they were sidewinding. And we think that largely because of what these myomeres would allow. By the way, when you fillet a fish, that's what you see. And later, next week, we're going to look at a perch. We'll do a detailed dissection there. And you see that there. So these myomeres have been around for over 500 million years. Something else that we have is our gills. At some point in development, all chordates have gills. And this is what we think Hycoela's gill system would have looked like. It looks a bit like a counter. I'm not sure if it's countercurrent, but more on that later. All right. So, and also good old mouth to anus. Speaking of anus, we have a post-anal tail, which is not something we've seen yet. Certainly don't have that in an earthworm or a nematode. So this is the easiest thing that we've seen so far that we can tag the name tail onto. And so we do, and we call it post-anal tail. So now I have a little video, a YouTube series, which I love. I've seen it a bunch of times. It's called Walking with Monsters, and it's Life Before Dinosaurs. So I'm gonna hit it. Before I do that, I'm gonna turn off my audio so there's no feedback. And I see there was a question. Which lab specifically do, oh no, okay, that's nothing new. All right, so I'm gonna turn off my audio for a moment. Okay, that looked like a technical difficulty. This time I will not mute myself and I'll see what happens. If sharing application with the video overloads and crashes me again, then I'll have to skip the videos. I have reduced the size though. But let's see here. All right. I do want to show you the video though, because I think it's cool. Let's see. Try this again.
This is Echinoderms. He's the size of your thumbnail, but he's an evolutionary giant. He's the first ever fish, our earliest known ancestor. He's unique because instead of having armor on the outside, he's tough inside. He's evolved a primitive backbone. He's the very first vertebrate, forerunner of all the backboned animals, from the dinosaur to the elephant to us. His flexible backbone makes him more maneuverable than spineless Anomalocaris. He can scavenge flesh, then dart away unharmed. Our tiny backboned ancestors have survived a sea of monsters, but there are still many more battles ahead. They must adapt or die. Evolution takes over. As millions of years pass, fish build on their basic design. The muscles around their backbone evolve into a powerful tail, and fins appear. They evolve a distinct head. He may not look like you or I, but this odd fish is becoming the food for our own planet.
Pretty cool, huh? Great. So that was Hycuella, or they call it Hycuelictes on there. So this is not a fossil. This is a living thing. It's called an Amphioxus, also known as a lancelet. So this is an extant living, meaning not, I'm just using synonyms for an extinct, not fossil, animal that has a lot of the same properties. And in lab, we pass around little cubes of plexiglass with these animals embedded in them, either whole or as a section. So you'll see something that looks very much like this at some point in your near future, and you may need to identify it by the genus. So you can call it an Amphioxus. You may be able to write lancelet, but this has the so this is Cephalocordata, and this is its anatomy. There's also a very large plastic model, like a 3-foot model of that looks very much like this. And in the past, you've had to identify like the nodal cord versus the dorsal nerve cord, that kind of thing. So biologists just cannot get enough of about this situation is that this living thing is so similar to this fossil thing 500 million years ago. We also love that we can watch a vertebrate develop or any chordate develop, and a vertebrate, and we'll see these five happening during its embryogenesis at some point. So there's this concept called ontogeny recapitulates phylogeny, and it doesn't always apply, but when it does, we go crazy. Ontogeny is life from the formation of a zygote until the end of life, and so that includes its development. And sometimes we see vestiges of organs that give us clues about our ancestry. So Amphioxus, lanceolate, cephalochordates, and those early fossils, Hycuella, and there's also this thing called Pica that looks very similar to that. We love it. So we love to teach about it, and we also can't resist testing you on it. So here are the parts of the body. The wheel organ functions like the corona of a rotifer. It creates a current, bringing particles in where there's some filter feeding action, and the pharyngeal slits. Those are involved in breathing and also the expulsion of old water. So in with the new water comes food and diffused oxygen, and out of the gill slits goes oxygen poor water and water that is more dilute, because it will have fewer particles, because some will have been trapped and eaten. And the endostyle starts that process. So inside this chamber, it's probably a little more, well, it's got to be a little more acidic or more basic than the water around it. And these are the gills. So we call these gills, and these are gill slits. And in a shark, say you can see those slits clearly. Of course, in a shark, there's a lot more separation between inner shark and environment. There's less of it in Amphioxus. I have seen these alive before in a lab. That I had a kooky undergrad advisor who loved to order strange animals and keep them around for us to look at, and I'll never forget it. And I want one. They're not that easy to get. So here's the notochord, and not to be confused with the dorsal nerve cord, which is always going to be smaller. And in a model, it will be positioned higher up, more dorsally. Like, it's very, very subtle, but I'm running my cursor over these little blue dots. This critter was stained. So this red is artificial. It's probably a stain called alizarin red. And you can see this clear shape where I'm tracing the cursor. It's not red, it's not blue, it's not anything. It's like basically a wooden rod, except it's not wood, it's whippy. And then above that, that this is a partially broken up stain job of the dorsal nerve cord. So there is a big difference in size. When you stain it, there's a difference in color, there's a difference in consistency. It comes from different cell layers. The notochord, well, actually, I'm not positive about that, so I'm gonna take a step back there. I'm not sure which germ layer the notochord comes from, but the dorsal nerve cord definitely comes from the ectoderm. Other attributes. Gonads, I don't really know what's going on there. That's okay, but there are males and females. No one's going to ask you about the gonads of an Amphioxus, I promise. It has the mouth to anus with the a hepatic cecum. Just know that this is a digestive organ. Not the first time we've seen the word cecum. Remember the cecum in the squid? We call it a cecum, not because it's homologous with the squid cecum, but because it is, like the cecum in a squid, just this extra pouch for digestion to go a step further. So a cecum is always going to be like an outpocketing of the stomach. There's always going to be a lot of nutrient absorption going on, and whatever is in the cecum will be mushier than what's in the stomach and what's up in the digestive system. So that's I don't think you're going to have to identify this, but it does help you make sense of what this is and not confuse you. You can yeah. So don't worry about the gonads, but if you see anything other than digestive tract, which will be the same color, then it's gonad. So, and remember, these are myomeres, post-anal tail. So next, let's talk about the beginning of a dorsal fin, which is kind of neat. Okay, so here's Amphioxus again. So you need to memorize these for lecture and lab. 5 distinct traits of chordates. So the dorsal tubular nerve cord that I told you about, that notochord, which is for structure and movement. The pharyngeal pouches or gill slits, endostyle for feeding. That's the thing that a derivative can be a thyroid gland. So endostyle evolved into a thyroid in some instances. And remember where it is. It's up here, which makes sense, close to the mouth, involved in feeding. And a post-anal tail, which is just wonderful for propulsion. So you must know these 5 things. You also need to know that there's an ad hoc, and I should count, but I think I've taught this concept for like 9 or 10 different course groups, and whenever they answer the question, like what are some distinct characteristics, you must add. I don't think everyone is this stingy, but I am. You have to say, or it's wrong. At some point, see how emotional I'm getting? At some point during development. So much emotion and smiley face. So you have to say that because I don't have a notochord, not me personally, and I doubt you do either. And I'm sure you don't have gill slits. I think I would have noticed, even during our few classes together. An endostyle, I think, would just disappear into your system, become nothing. You know what? Maybe some of you do have a post-anal tail. I didn't look that close, to be honest. We still do have a dorsal nerve cord. Actually, no. You know what? All these things are gone by the time vertebrates become by the time humans reach the end of their development. So at some point during development, where it's wrong. Okay. So I want to show you a new video. So this is about the notochord. So this is Amphioxus. Sometimes it embeds itself in the sand and filter feeds with its upper body exposed. This is attention to that stiff notochord. And during human embryogenesis, we begin with the notochord. That's the pharyngeal gill pouch. Those are those mesomeres, the myomeres, sorry. Remember the nerve cord is on top. And we get something nice from this notochord, which is a vertebral column. So this doesn't have a notochord, this elephant, but it did. This alligator, same thing. The herpetologist in me wants to tell you the difference between an alligator and a crocodile, but knows that I have to do my job too. I am a herpetologist. I don't know if I haven't mentioned that in a while. I study amphibians and reptiles first and foremost. So back to those 5 chordate characteristics, which I'm drilling because you're going to get tested on it for sure, and you'll be cross-tested on it for sure, lecture and lab. Also, this has a presence on the MCAT and also the bio what's called as the SAT2s. Oh, the bio GRE. Not many people take the bio GRE, but this is would be on there. So here's the 5 chordate characteristics: pharyngeal gill slits at some point during development. We actually have pouches in the human embryo. I don't know. I don't think they're actually slits, but we do have pharyngeal pouches. I don't think the slits actually form, but I could be wrong. That endostyle, which can evolve into a thyroid gland, it's for digestion. That muscular postanal tail for movement, coordination, and whoa, okay, so now I'm leaving cephalochordata. So if you have questions about this group, you should write it down. But now I'm moving out, and I meant to have a tree to show you first. So this doesn't seem so random, but this is where. Oh wait, 1 second. Now we're in here. I did have a slide there, and I don't know what happened to it. Can't just jump right in. All right, so now we're leaving chordata and moving into hemichordata. And we would not have live specimens of this in lab, but we do have some preserved specimens. So hemichordates are more closely related to the echinoderms than they are to chordates. You should know that. So know this arrangement. Echinoderms, or you should know echinoderm, hemichordate. I don't think you have to worry about the relationship between Enteronusta, pterobranchia, and echinodermata. Just know that these 2 are together and they're hemichordates. So it looks like a lot of information, but if you start like just doodling it in on napkins and margins when you're watching TV. Just making this tree a couple of times and you'll be fine. And these words won't seem so alien. I'll say them all, so you'll have heard them said at least once. Hearing them, knowing how they sound is helpful. Deuterostomia, which we understand what that is. That's this whole clade, characterized by mouth not derived from blastopore, and pterocoeli, which is when the gut, which is when the gut forms from an out pocket of the inner layer. So this has to do with how the gut forms. And ptero, presence of gill slits in the pharynx at some point during development, remember, and radial determinate cleavage. So that's for deuterostomia. Then chordata, we just discussed that. There are actually 5 here. Remember, postnasal tail, notochord, endostylar thyroid, dorsal hollow nerve cord, and the pharyngeal gill slits are already included in there. So those are the 5. Now moving out, Xenoturbellida, which is the peanut worm, and I don't have a slide on it, and I don't think we discuss it. But if you want to YouTube peanut worm, I recommend it. They're just weird. But Xenoturbellida, peanut worm. I don't know how diverse they are. They're marine. They're all marine. I would of course like one as a pet and can't have one. So next, Ambulacraria. So Ambulacraria does not include the peanut worm. The peanut worm. See, it looks like a peanut. Ambulacraria includes echinodermata and hemichordata. So echinoderms, those are the starfishes, the sea urchins, the crinoids, and the ophiuroideans, which are called which are not, they're not true sea stars. We'll look at those later. So Pteroneusta, Pterobranchia. So I've said all the names. And now I'm going to talk about Pteroneusta a little bit. This is the acorn worm. And what these have in common, something called a buccal diverticulum, which is exactly what it sounds like. Buccal is always, or some people pronounce it buccal. It's going to be like the mouth. In humans, like a buccal swab, you take it from the cheek. Diverticulum, that means there's some sort of septum or divergence in there, a separation in the mouth area. So it has that. And mesosolemic ducts, and I'll actually show you those, which is also what it sounds like. Meso in the middle, coelemic, so body cavity, and ducts, and you can actually see them right here on the Pteroneusta. So it's a very weird looking animal, Pteroneusta, the acorn worm. This one, its genus is Saccoglossus apparently. So they have, a little anatomy. They have a proboscis. So this is the mouth. It doesn't eat with it. The mouth is here. This thing, the proboscis, it uses to dig into the ocean bottom, and it pumps a little bit up and down, and that does help move food into the mouth. So here's the mouth. It's just such a strange animal. We look at it on the outside, you go, okay, it's a worm, but I mean, compare this to the earthworm. The anatomy is so incredibly different, and it is just far more complex, arguably, in the acorn worm. So a worm isn't a worm isn't a worm. One thing you can learn from this class is that there are a lot of long, wormy-looking creatures that just the like body plan works over and over and over and over and over again. And any worm-like thing could be any one of so many different animals that diverged hundreds of millions of years ago. So that's one thing about organismal biology. So this proboscis is muscle. It's the, I guess if you were going to eat any part of this, not that I would, you'd want to eat the proboscis. That's the muscly part. It has a real heart with pumping. Here's its pharynx, which is the where food is going to come in, and also gill slits for breathing. So yeah, so the mouth doesn't have any special features. It looks sort of like no teeth, just sort of gummy lips, and the collar is muscular and that moves. And look, it has a nerve, a collar nerve cord. So I have a video of this peanut worm. I'm gonna play it. So here it is digging with its proboscis, and notice it said 1. I want to point something out here. So this will help you remember that pterobranchia exists. The acorn worm is one of our closest invertebrate relatives. Pterobranchia. So hemichordata is closest to chordata, and hemichordata is in Pteronusta plus pterobranchia. So that's why it says one of. And there it is digging with its proboscis. There's its collar. I want one so bad. So this little video clip by University of Washington, their fascination with this animal is its regenerative properties, which really exceed what an earthworm can do, because I think it might be fast, comparatively fast, and also the anatomy that it regenerates is much more complex than earthworm anatomy. So this is an accelerated image sequence of the regeneration, and I've got to show it to you again because of how cool it is and because it went very quickly, because it's a fast process, a lot faster than you might expect. So there are researchers who are very, very into this because of the regenerative aspect. So because it's a rather close relative, we share a lot of genetic material, which usually means that what you discover in an animal that's closely related to you is more applicable. So here's day 0. They cut its whole head off. And so that's day 0. Another reason why I'm showing you this video is because you can see the buccal diverticulum. Something's wrong. Hey. Okay, I just checked to make sure the line was still active. Okay. I'm determined to show this to you guys. Baser. All right, my determination ends here. I'm afraid to crash it. But there you have it. Amazing regenerative properties. I'll watch it one more time. So just watch closely when they show the accelerated regeneration sequence, because I'm not gonna pause it. Okay. So I've showed it to you. So here's how the feeding works with the collar. The collar moves back. That opens the mouth. That also creates a little bit of a current. And this is mouth occluded. The collar closes. So the collar moves. It's very sensitive. It has lots of nerve endings in it. And lots and lots and lots of small muscles. Oh, and this is important too. Currents created by cilia on the proboscis. So you could be shown a picture of it in lab, or a specimen in a jar. So here's the other hemichordate, pterobranchia. Ptero is going to be a reference to wings. We see ptera, T-E-R, quite a bit in biological and scientific nomenclature. And branchia is usually a reference to something respiratory. Probably not arms, because then you lose the N and you have brack, like brachy. That's usually arms, but branchi probably has something to do with oxygen. And here it does. So wing gills. It's basically arms that are wing-like, and they have the ability to absorb oxygen. So the pterobranchs are sedentary for the most part. So they don't move around very much. They have a sedentary lifestyle, but they can move. And I think that the larvae move around more. And that is important to mention, because one of the characteristics of animal, in order to be an animal, there must be some motility at some point during its life cycle. So they're sedentary, but they do have a point where they have moved. Plants, not so much. So this is one criterion that makes plants and animals different. But again, by and large, sedentary. So this is the proboscis, and there's a proboscis in the acorn worm as well. And there's also a type of collar that moves in tandem with the proboscis, and opens up to the mouth. So it's kind of like an acorn worm in that way, but it's where is it breathing? Blackstone, retracted zone, rect tube. Groove. We're going to revisit this because I don't want to mislead you. I was under the impression that there was some oxygen absorption in these tentacles, but now I'm not so sure. And so we're going to revisit that. I'm actually writing a note right now. But you can see the one reason why this is called a urochordate is because it has the U body shape. Mouth, U, out the anus. Mouth, U, out the anus. There's another creature we looked at, and I'm not going to tell you what it is. It's a personal little quiz question for you, but we recently looked at a creature that has a similar setup, and I drew on it with the highlighter to show you, and it's on the PDF, the lab manual PDF for one of the chapters. So just be aware of the other animal that has the U. So here's a live one. I just love these little things. These are the terebrants, the kits, and they're so diverse. They are pretty tough. I have kept a few of them as pets. I tried to. They don't live very long in my tanks, probably because of the same reason why my sponges don't live too long. There isn't enough stuff in the water for them to eat. So it's kind of a bummer. They're very diverse. They're strictly marine. The tunic is a reference to it's kind of leathery, and only the larval form bears all the chordate hallmarks. So here again we're seeing it has those 5 things at some point during its development. So I don't see any kind of gill slit or anything like that, but it would have had one as a larva. So here's another look at its body. Here's the incurrent siphon. Okay, so I'm so sorry, it does not breathe with the arms. That thing's revisited. We're done. It's absorbing oxygen on the inside, which really does now make a little more sense. And it may have branch-like tentacles. All right. Yep. Stomach, heart, gonads. It's got it all. So here's a free-swimming tunicate larva, just demonstrating that they, early on in development, they have the notochord, they have the nerve cord, they're free-swimming, so they're moving on their own. They have an endostyle as well. So chordate, it's really checking out. And then it finds a space for it. It metamorphoses. Notochord degenerates because if it doesn't need it anymore, and neither do humans need their notochords once the spine is developed, so that it degenerates there. The notochord doesn't become the spine, but it does provide support for when the spine eventually develops. So this is just another example of at some point during development. And I wanted to show you, this video is kind of neat.
Discovered something very unusual. We went off down with another couple of guys on the boat.
Sorry about the sound quality.
What Mick Barron saw next took him completely by surprise. Well, I jumped in the water first, and then my camera was passed down to me. While I was setting it up, I saw this strange animal about 30 metres away. I thought, wow, what is this? So I yelled out to the other 2 guys on the boat, come on guys, get in here, you've got something here you'll never see again in your life. enormous tube-shaped structure was hanging in the water. It was about 10
Imagine
meters
that.
long. When I touched it, it didn't actually react. It just sort of felt like a soft jelly-like substance. And strangest of all, the tube was glowing with an eerie blue light. Was its origin even alien? I've been there for quite a long time, so 40 years. So when I saw this start to blow up, it was a pretty unusual experience. So what was this gigantic glowing tube? And was it even alive? When it comes to the ocean, we've barely scratched the surface. Here,
Okay.
even these bizarre creatures have features that we can recognize. Eyes, limbs, teeth. The thing that the divers encountered had none of these. But there is another possibility. You see, some marine animals live together, forming a much bigger organism. Take coral reefs. Corals are formed by colonies of creatures known as polyps. These tiny animals build a hard exoskeleton around them. And living together in their billions, they form some of the world's largest and most spectacular structures. Like the Great Barrier Reef. And it's not just corals that take to communal living. Our giant tube is actually a colony of tiny animals, known as a pyrosome.
Hey. Yeah, so that thing is predatory, actually, the pyrosome. Okay. So we've talked about some hemichordates and chordates, hemichordates, this thing that I just showed you, the sea scalp, aka the tunicate. This, just to be clear, they belong with the they're in here, not there. So Pterobranchia and Interanusta are they're worms basically, but that sea salp, the tunicate is in here, even though it's a urochordate. There would be this other branch not shown, but we do have some sea salps in lab, and I'm going to go over that again tomorrow. So okay, so now I'm going to keep going and talk about echinodermata, which we focus on big time in lab because we do a whole starfish dissection, and the instructors demonstrate a sea urchin dissection. We usually don't have a whole heck of a lot of them, so that's why that is more of a group activity. But y'all would have your own starfish, and you do a dissection, so you will be tested pretty heavily on starfish anatomy. You'll have at least one question during the practical, but you'll probably have two. And but it's possible you'll only have one. That's its strictly anatomy, which means you have to learn all of the relevant anatomy in order to get one question right. And the same may be true for the model of the giant lancelet. So, but here are the characteristics. Endoskeleton, this echinodermata now. It's just wildly different from everything we've just looked at. It's taking a huge step away, more than 500 million years. It's wild. So they have an endoskeleton of what's called stereome ossicles. Whenever you see ossicles, you know there's going to be some sort of little bony hard thing. So they have a bit of an endoskeleton, and they have spines of some sort on their skin, and they could be derived into little knobs or plates. So they have pentaradiality, and penta, like a pentagram, 5 points. So so an echinoderm either has 5 slices or segments, or it's a multiple of 5, or it's just 5 at some point during its development. And that is important to remember because there are some starfishes that have like 40 arms. Maybe not 40, but way more than 5. So pentaradiality isn't really the same thing. It has 5 arms, but pentaradiality, characteristic of echinodermata, kind of easy to remember, and no gill slits at all whatsoever, erased. So now the rest of this lecture is all echinoderms, and we're making pretty good time, or we're gonna have time left over, I think, so I can maybe go back and cover some other things. So, funny thing about echinoderms, they have, well, one thing about deuterostomes is that they are, and protostomes for all or the most part, is they've got bilateral symmetry. In other words, they're not pizza slices. If you cut them one way versus another way, you won't necessarily get the same parts. So that's bilateral symmetry. I would read, please read about lateral and bilateral symmetry in terms of biology, because of all the exceptions that exist and the ad hoc explanations that you will probably miss if you only learn it once or only listen to lecture. So I would like you to do the homework and understand what bilateral symmetry is, and what has them when. So do that for yourselves. But pentaradiality, it's a derived feature for echinoderms, but their larva is bilaterally symmetrical. See how, like, if you slice it this way, slice it that way, even slices, different directions, different parts. Probably wouldn't work out so well for the animal if it was cut in half. You can cut this way and get equal parts, but you can't cut that way and get equal parts. But that isn't the case for the adults. So the larvae have lateral symmetry. So this does squeeze them into the bilateral symmetry at some point in their lives category. But the adults are pentaradial. If the ontogeny recapitulating phylogeny concept is in effect, and you can't always be sure, but sometimes, then we could, there's a hypothesis, and there's an alternate hypothesis too, that the ancestor of the pentaradial adult was something like this, but they've retained the bilaterally symmetrical larva, which is great for dispersal. Really, these things are more motile. Imagine if this thing just had tiny little tentacle baby starfish that could only crawl around, or that were too heavy to move in the current. And it's better for populations and establishment and colonization of new areas, and just surviving as a species if the ability to disperse exists. Oftentimes, that's usually true. There you have it. And the larvae are pretty interesting, I think. So those three characteristics of echinoderms: the endoskeleton, pentaradiality, no gill slits at all. So some more things about them. Can't osmoregulate. Osmoregulating is dealing with salt. The body, it can't have too much salt, and it can't have too little. If you put it in fresh water, it will die very quickly because it will overindulge on fresh water. Basically, it's equipped to make the most of a low salt-free water environment. So when placed into a very low salinity solution, they basically it will let their cells explode with water. They become oversaturated. Echinoderms have all sorts of lifestyles. Most of them do well. The ones I handle most do. They graze, they scavenge, and they'll predate, given the opportunity. And I added none are parasitic. Isn't that nice? Because usually, like, I don't know of another example of a clade that is this large, diverse, and old, ancient old, and not one of them has gone the way of the parasite. That's just unbelievable to me. Parasitism evolves over and over and over again. Just one thing leads to another, and something becomes parasitic. But that hasn't happened in echinoderms, not once. I don't know. That's mind blowing to me. I can physically think of ways where a parasitic relationship could start, and they just haven't for whatever reason. So I don't know. That's a mystery to me. But they've got that pentaradial symmetry in the adults. Bilaterally circular larva. We just went over that. I think we have a slide in class, or at least we did at the last place I taught, of starfish larvae, and they look pretty wild, and they do make up part of the zooplankton community, which is super important. So you need lots of larvae in a lot of in certain systems, because things that eat plankton need them. So echinoderms provide new food for other things, as adults too. So all echinoderms has some sort of external spine or protuberance that can manifest as like leathery skin. So echino, this is a reference to a spine. There's this other animal called an echidna, and it's spiny, the way that a hedgehog is. So echino, this is spine, dermata, derm, of course, derma is skin. And so when this goes to the dermatologist knows just how to handle its spines or knobs or whatever it has, depending on what type of echinoderm it is. So that's a characteristic there. Also, they have that calcareous endoskeleton, which I showed you right there, and that's the benefit of opening up a sea urchin in lab and a starfish in lab, because you can see that they're not so they're pretty tough, and it's just very, very helpful for them because they're often very, very exposed. I think they would be eaten left and right if they didn't have the endoskeleton and the leathery skin. Also, it keeps them, it gives them structure, and they need that because they're largely hydrostatic in other parts of their body. And sometimes they eat things while they're still alive, so it helps to have a strong body to contain them. They have pedicellariae. I'll say it slower. Pedicellariae. No, it's plural because it has the AE, ped, feet. And that's a reference to tube feet. And even sea urchins, which are spiny, have tube feet. So you should probably learn this word because you may get tested on, it'll either point to one directly on a whole specimen, or it could be a cross section and it'll point to a cross section of an arm, and an arrow may designate an individual pedicellaria, and you'll need to recognize it. And it's how they move and grab things, and they're very sensitive, and they are so fun to watch. So they also have dermal branchii, and I'm honestly not entirely, now I have to Google that. What is dermal branchii? Is it what it sounds like? I mean, I could make a guess, but it's going to have something to do with breathing. Let's see here. Dermal branchii. I want to make sure I get it right for you. Yeah, it is respiratory, sure, because it says branchii. Skin gills. Okay. So they're projections of the coelom that I guess cut. So slits, I suppose. All right, that makes sense. They're pores. And we'll be able to see them in slides. Great. I learned something a little better today. Dermal branchii, extensions of the coelom. That's how they get their oxygen from the water. Okay, and that slide was supposed to be earlier. But anyway, so yeah, please ignore the previous. We're in echinodermata. That slide was supposed to be earlier on. So here we are now, echinodermata, and these are the four main groups. Well, five, I guess crinoids are outgroups, and we don't look at them in lab, I don't believe. But if you fossils, and you're in a place where it was marine stratigraphy at some point, if that's the where you're foraging for a fossil, you find little stacks of rings sometimes, and sometimes they're shark spines, and oftentimes though they're crinoids. So this is a common fossil to find in marine stratigraphy, and it is not. Oh, and it is an echinoderm. But you don't need to know this word, palmatozoa. I'll show you a crinoid that they're also called baskets, sea baskets sometimes. They're pretty neat. I've never seen one in the wild, even though I scuba dive. Echinodermata, back to where we were. They have an oral surface against the substratum, so that simply means lower down on its body, facing the bottom. So underneath it. So this is dorsal. So it's basically face down. So that little, if you've seen Finding Nemo, the little cartoon starfish with its face against the glass is accurate. It has suckers on the podia, so those little tube feet, and it has, and this is a word you got to know, and it's one of the most standout organs the outside of an echinoderm. It's called the madreporite. It's on the aboral surface. I'm so sorry. Earlier I said dorsal. I'm supposed to say aboral with the starfish, because this isn't really its back. It's a different. We use a different body plan term scheme when we're talking about starfish and other things that are kind of similar. Aboral surface. My apologies for it's better not to say dorsal. But if I don't think anyone will get too upset with you if you do. But aboral is proper. Aboral shows you really know what you're talking about. So they're attached to the substratum by the aboral surface. The madreporite. That's what I was talking about. So all echinoderms have a special pore. It's basically a circular sponge that's embedded somewhere on its aboral surface, and it controls water coming in and coming into it. It's like a valve, a combination sponge and valve. It's porous. It's very cool, and it's easy to spot. And we'll go over that in detail. But presence of a madreporite. The asteroideans, the ophiuroideans, the echinoideans, and the holothuroideans. They all have those. So moving up, ambulacral grooves. This is another word you need in your bank. In addition to, so what do we have so far? Words you should absolutely have to succeed in your life, or at least for the next few weeks. The endoskeletal plates, or ossicles, you can say. Ambulacral grooves, and those, I'm going to go into more detail about those. And they have to do with their limbs, if you will. Pentaradial, that's something you need to know. 5 ambulacral, which is related to the pentaradial symmetry. The ambulacral grooves are the arches underneath here. So we'll look at those. What else? Podia, which are the feet. You should know that word. Madreporite, you must have that word. So now moving up. So now this does not include starfish. This characteristic, closed ambulacral grooves. And that's very obvious. We're going to look at when we look at these 3 groups up close. The openings that you see the grooves, you could fill it with something. You could fill it with Jello or pudding or ice cream, or I don't know why you would. But there's volume, there's space under here, under the arms, and that is not the case with these 3. It's all closed up. So that evolved in the ancestor of these 3 groups, closed ambulacral grooves. So now I'm going to go to the left because I love these animals, the Ophiuroideans. They are so people who are ophidiophobic are afraid of snakes, and this is called ophiuroidea. And it means snake-like, like a snakeoid, snake-type thing. So this is a reference to the snake arms. They're called brittle stars. So these move very differently from true sea stars. And I'll show you a video of that. The madreporite is underneath instead of on top, so that's important. Still as a madreporite, it's just down here. In the starfish, it's up top. So that's an interesting evolutionary event. So another thing that happened along the way was loss of podial suckers. So those pedicularia are not there anymore, and that's because they have a different way of moving. Okay, so now moving out to, so good to know, you can see this here. You could get tested on this. The sea urchins are more closely related to sea cucumbers. Neither of them are to these. You can also see that the brittle stars are more closely related to sea urchins than they are to sea stars. So that's the harder pill to swallow. So that might be the one to focus on. But you should know this arrangement. The cucumber, the urchin together, the cucumber urchin plus brittle, and then the outgroup to those are the starfish. It would help to remember sea stars are all on the outside, and that's easiest to recognize because they still have those grooves under their arms. So that makes them immediately different. You can think of these two as having less personality because they don't move as much. So that sort of, you can think of them as being united that way if you want to. And then this is their wacky cousin who moves around a lot and who has retained this basic shape. Now, I'm going to cover this really quick. Echinoidea. So this is the nominal group where they looked at this and decided we're gonna call this an echinoderm, and then these have these in common. But this is what's called the nominal group. It carries the stem word of the other taxonomy. So echinoidea, the spiniest of the spiny-skinned animals, the sea urchin. They still have tube feet. Look at them; they're awesome. They have fused skeletal ossicles, and they're pretty rigid. So then its sister, the sea cucumber, Holothroidea, which I think they're very interesting. Oh, I forgot to note the things uniting them: extension of ambulacral grooves from oral to aboral pore. I don't know how obvious that'll be when we look at it, but the aboral surface is highly reduced to the region around this, which shows, in other words, like it's got this nice big flat aboral surface here, nice big flat surface here, but you can think of it as its sides sort of rolling up and expanding and becoming more complex, while this whole surface just becomes very small little bit. So lots of expansion and derivation and new features here, and all of this shrinks down and becomes rather small. So they have that. So now in the here's Holothroidea. Obviously, elongation of this axis. It's a bundle anymore. It has some linearity to it. So it's elongated. It has, yeah. So that's the most obvious difference. They have, and this is I think the weirdest thing about them is that they have an internal madreporite. So that makes the whole understanding what the madreport is more complicated, which is why it's best to study it first in these animals, and then you can think in terms of, okay, so now it's rolled up on the inside. I guess that works too. So keep in mind, I saved this for last because of that. And we do examine sea cucumbers in lab. We don't dissect them, although I once taught at a place where we did dissect them, and that was actually a real trip. There's a lot to see inside of them. They're pretty neat. And the ossicles are reduced. They're not as spiny. But that's a general statement because some of them are they have derived spine-like features, but overall those ossicles are shrunken down a little bit. So I've just talked an awful lot about this slide, but there's an awful lot to digest here. And I believe I'm going to cover these features again in the next slides in more detail. And I'm going to try and do it justice because they're really cool animals. They're all marine, by the way. They're crucial to the food web and to water chemistry. They're massively important to life on Earth. And here are just a couple of here's some diversity. So you should be able to identify what each of these are. They're either holothuroidians, they're ophiuroidians, they're asteroidians, or they are, I just named four things, or they're crinoids. So you should be able to know that. Echinoids, which is this. So this is echinoidea. This is an urchin. This is an asteroidea. This is a true sea star. This is another echinoidian. This is an urchin. This is an ophiuroidian. This is a brittle star. This is a crinoid. This is a holothuroidian, a sea cucumber. And so you should be able to do that, and then also know how they relate to each other on the tree. So you should be able to ID them that way and then associate them this way. So I'm gonna hold this for a second. So if you rewatch this video, you can test yourself. Okay. So I want to show you a starfish eating a mussel. And they put the camera in the mussel. Very cool. So they're moving 2 feet, are hydrostatic. They decide to move where they move. Oops. Oh shoot! I forgot I should not pause when I have these videos. I pre-record them so that you don't have to see ads and we can stay in the same application. But that does limit flexibility. Anyway, so here you can see its spiny skin. It's searching little tubercle feet. There's its mussel that it wants to eat, that I want to eat. Frankly, I love mussels. So it's making a choice. It's picking a mussel. It found one that's open enough. It looks at it. And then they put the camera inside the mussel that it chose. And here are its tube feet pushing into the mussel, and digestive glands being pushed into the mussel. It is eating the mussel. The mushy part is the starfish, and it is eviscerating itself, sort of. Its stomach, a gastric organ is coming out of its body and is inserted into the mussel. So it is trying to eat from the inside. It's reaching into the mussel with its digestive system and beginning. And if it's successful, the mussel is going to loosen because it's being eaten. Now it's withdrawing. And now the muscle's gone. It ate it. So sometimes a starfish will choose its prey and position itself on it for a long time, pulling it open further, slowly, 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 using its tube feet and muscles, and its own muscles. They'll eat clams. They'll eat all kinds of things: clams, mussels, oysters. They'll eat worms a lot of the time. They eat algae. They'll eat kelp. Um, I'm pretty sure if you like put a turkey sandwich underwater, it would start to eat the turkey. It may also eat the lettuce on the turkey sandwich if you have that. It depends on the species, but by and large, I don't think they're really specialists. Anyway, so this slide is super important because there's a big model in the lab practical that is almost exactly this. And I think there's a separate model even that is just this. And there are also slides, cross sections that show this. So you need to know, and seriously, there may only be one question that tests you on your knowledge of this, but you need to know all of it in order to get it right. So that is just how we do. But we'll start from, let's start from the tube feet. So that these are the pedicellariae, the tube feet, and they can each move independently. And it's a hydrostatic process. So water will be flushed into the tube. So it's like a sleeve, and they extend. And then they don't use slime or anything like that. They just form a suction, and they form a tiny vacuum, the way that you would if you had like a toilet plunger. Essentially, they're like little toilet plungers, and they line the animal. And this is the ambulacral groove right here, by the way. So this is on the undersurface of the starfish. So the ampulla, that's an individual origin point of each tube foot. So so if you remember, I was showing you, if you attended the review last week, I showed you a lily plant, and then you've got the ovary inside of the ovule, basically like one larger piece, and within that piece there are other pieces, and it's like that with the tube feet. So know that tube feet, ampulla, pedicellariae are linked terms in that sense. So the ampulla is just the part. The tube feet, it's the whole thing, really. Or you can think of it as just one of the extensions. And they're all connected by the central canal. And here's the sieve plate, which is also called the madreporite. So this is what happens. There are a couple things happening all at once in shared space in the starfish. So I'm going to walk you through that because this is half the lab, basically. You spend a great deal of the lab portion dissecting a starfish and learning these things. So that's why I'm going into so much detail. So the sieve plate, it's also the madreporite is over it, or it's a synonym for it, I'm not sure. And this is where water comes in. So there is a ring, but it's hollow. and it can fill with water. And when it flushes with water, all of these extend. If a lot of water is released, the starfish will be sort of small and shrunken and probably not moving around. Maybe it's feeling a little bit vulnerable, or it's resting. So there's the greater control of all the water that comes in, and there's also individualized control over each foot and each arm. So there are, this is the main valve where everything, all the water goes in and out, that has to do with movement of the tube feet. And then there's lesser tiers of control after that. So they've got like a pretty complicated nervous system going on, and it's a bit like a spider, or a crab in a way, where, or an octopus, where there's a lot of reactivity that's somewhat independent and localized. So they have like localized nervous activity, and pretty strong chemosensory. Starfish can taste when something it wants to eat is in the water, and it can sense. It's a little bit photosensitive. That the eye spot doesn't really form a picture, but it can distinguish between light and dark, which I guess it has its uses. One of them could be that if an animal is over it, like if the fish is hovering over it, or a seal or something, it can sense the change, the sudden change. Also, maybe it behaves differently at night. So this is the tube feet system, radial canal, tube feet. So know all those. So on top of that are digestive glands, and often, depending on where it is in its reproductive cycle, like it could be seasonal. Either most of the arm matter will be digestive gland, or sometimes it's overshadowed by gonads. But the digestive gland is on top of the gonads. The hardest part of the starfish dissection for anybody usually is sometimes the gonads are hard to distinguish from the digestive glands when you're looking at the specimen. Here it's very obvious. But the digestive glands have a much easier to spot connection to the digestive glands in the other arms, and to this, which is called the pyloric stomach. So this is a term that you need. You need the pyloric stomach and the cardiac stomach. The cardiac stomach, I'm going to double check here right now, but I'm pretty sure that is the thing that extends. But I'm pretty sure the pyloric stomach is going to be internal always, and it will pump a little bit. One second. Okay, let's see here. Okay, yes. So it is the most logical looking thing, which is that the cardiac stomach is what extends into the thing that it's eating, if it extends it. So food first comes into contact with the cardiac stomach, and then there's a a constriction that separates it from the pyloric stomach, and we can't see it. It's underneath. So this billowy, mushy thing, tons of digestive enzymes in it. So this is the thing that's dangerous to its prey, because it enters it and it fires enzymes to the point that the prey animal succumbs, and it can't defend itself anymore. So food enters here. So, when it's enveloped, it enters the pyloric stomach, and we can't see that connection. That's okay. And nutrients are distributed, and the glands feed into here, by the way. Let's see. Yeah, so sometimes food is not fully processed when it enters the arms. And when it enters the arms, that's where a lot of the final digestion occurs. So it's a strange system. And then if it's going to cycle back and go out the anus. So I know that's a little bit complicated, but I'll to 1, 2, 3, 4. Step 1 is the cardiac stomach. Step 2 is pyloric stomach. Step 3 is is in the arms, and step 4 is out the anus. Done. So you should know that the sieve plate and the anus are both on the aboral surfaces. And then the gonads are usually tucked pretty tight against the inner walls. There's a lot of space in here, by the way. It really sometimes just seems like a leathery bag. Okay, so I've just talked a lot about this because you're going to get tested on this somehow. Oh, and the gonads, I'm not entirely sure where the gametes come out, to be honest. But so let's have another look at this. The real starfish, and this is so well done. This figure, it's from Journal of Anatomy, and just glad that this exists. So you should study this. You should really study this. So PC, that's pyloric cecum, and remember it's higher up than the gonad because it can be tough to differentiate there. Here it's a fresh animal, and I do see a little bit of jelly-like stuff, which could be gonad. In fact, I think this here, that is gonad, and over it is the pyloric, is extended pyloric cecum. And here it's very obvious. By the way, these are those little ossicles. I said it has the echinoderms have endoskeletons to some degree. Most of them do. And that's what these are. These little plates right there, those are the ossicles. And among them are the tube feet, TF tube feet. Outer body wall, the pedicelaria, those are the individuals ones, and this is for breathing, these openings. And the madreporite is not shown well here. So great image. I would study it. Here's another section of an arm. In case you couldn't tell, but here they make it very clear. This is a section of an arm. This is also a section of an arm. And it's good to study the same anatomy using different images, because you have to make the connections, and it really helps you understand well. So that's why I have a few. Hepatic cecum. I think we can interchange that with pyloric cecum here. I think that's okay. Here is the spine of the skin. Gonad, lower down, gooier, not as distinctive. Ampulla, tube feet, and here's that ossicles, the ambulacral ossicles. So this is the ambulacral groove right here. So a lot of the starfish is digestive material. And they really get bigger when they eat something. And this is the madreporite. This is that small opening that is so important. It can't move its tube feet. It can't move its arms without the ability to have an influx of water and a release of water, and it controls that through here. And it's small, but it looks different, and it's easy to spot. It's the circle, the distinguished looking circle on the aboral surface of the asteroidean. That's the madreporite. So here is yet another image to help you learn the ins and out of the starfish arm. So this is also a cross section of the arm. And here again, the pyloric cecum, it's rather wiggly. Up top, here are the tube feet, the ampulla, that would be the bulb that controls that tube foot. So remember, this and this are connected. And let's see what else we have. Oh, so this is for breathing. This is for oxygen diffusion. There are little pores. Let's see here. Oh, another ampulla. Here you can see the connection. It's a little more obvious. So think about this being full of water and squeezing and constricting. That's going to push water out to the tube feet. And alternatively, the tube feet can squeeze and constrict and reduce its volume, and that water is going to go back up into the ampulla. So it's sometimes just a flushing, tossing back and forth of the water that extends the tube feet. Kind of neat. But the tube feet has individual control at the ends, and those are the pedicularia right here. And that's how it forms that seal. So let's see. Why did I have both of those? Oh, just I wanted to show you a bigger one to look at. It's just really neat. Okay, so now we're moving out of Asteroidea, and we're in Echinoidea. Let's see, we've got 25 minutes. I like it. I like it. Echinoidea. So I'm skipping over Ophiuroidea for now, but we're going back to it. So echinoid, actually, it makes sense to go in order. Okay, I heard a message. So by the way, whenever I hear someone send a message, I go flying out of the app to see what it is. So your quiz is at 11:45. Oh my goodness. Okay, thank you, Ariel. Glad I checked. All right, oh shoot. I'm gonna go for 5 more minutes. I'm sorry too. All right, let's see here. Okay, I can wrap this up in 5, and it's a good thing I'm doing another review tomorrow at 1 o'clock. Oh no, not from the beginning. From the current slide, please. Nope, negative. Well, that's too bad then. Hopefully you all are available again tomorrow at 1. Okay. So Ophiuroidea, the brittle star, they move. They're the most active by far of the echinoderms. They move by bending jointed muscular arms rather than walking on 2 feet. And I have a neat little video that shows that. They have no grooves underneath them. They have no suckers. They move like this. I've had them as pets. I enjoy them. There it is. So that's why they have a reference to snakes in their name, Ophiuroidea, because of how their arms move. And this is not sped up. They move very quickly. People are passionate about their animals. So you can see the pulsing of their main body becomes very large after they've had a meal. They can be pretty aggressive. They're not poisonous. They have, and those are spines. Those are not suckers. They don't have, they have interlocking joints, which is very evocative of the crinoids, which is that other type of echinoderm. I just love them. I see how, I find that creepy, but I get very excited when I see brittle stars. And that's a little octopus. He just doesn't know where it's at right now. There it goes. It can't deal. Regenerative properties. There it is, regrowing some arms. They don't regenerate nearly as fast as that acorn worm. All right, I need to let you go because you have your quiz. But we had a couple more slides, urchins. What we didn't cover today were sea urchins, the anatomical details, and we did not cover the holothuroideans, the sea cucumbers. So I hope you're available tomorrow. Yeah, so 1 o'clock tomorrow, tune in for a review session. Good luck on your quiz, everybody. You are welcome. And Nava, if you could email me an alternative email address, and I'll CC you on something to the Blackboard people, because you need to be able to have contact whenever you want it. There may be a glitch specific to your email. Okay, thanks. Good luck on your quiz, everyone. See you tomorrow at 1. Hopefully you can join.